Okay, we're going to be talking about meiosis, or some people pronounce it meiosis. So there is a video clip here linked to this. So if you will click on that, watch that in Ed Puzzle, and then if you'll please come back. So when we're talking about meiosis or meiosis, we are talking about cell division, but the reason for the cell division and the genetic makeup of this cell division is different from mitosis. But the actual processes that occur in the cell division are the same. So if you know the actions of interphase, pro, meta, ana, and telophase, then you know the actions in meiosis. It's just that the number of times that a cell goes through those stages and the ending genetic makeup, including the genetic number, will be different. So in meiosis or meiosis, the cells that are produced are going to be genetically unique. They're going to be genetically unique to the original parent cell, and they're going to be genetically unique to each other. The resulting cells are what are called haploid cells, but I like to refer to them as haploid. The cells in mitosis were diploid or diploid. Unlike mitosis, which occurs throughout an organism's life for growth and for repair, meiosis only occurs during certain stages in an organism's life cycle, and that's during the time periods when a person will reproduce. So, from puberty to menopause, we could say, if we're talking about females. And then when we're talking about what type of reproduction, we're not talking about the creation of new skin cells or muscle cells. We're talking about the production of what are called gametes, egg and sperm cells. So this is involved in sexual reproduction. Now, we do need to talk a little bit about chromosome number before we get into the actual process of meiosis, the actual cell division. So what you're looking at here, let's see here and here, you're looking at human karyotypes. And a karyotype is a chart to look at the chromosomes of an individual. And we've got two different karyotype charts here. The first one is a diploid, or of a diploid cell, that is represented by 2N. And these cells are somatic, they're body cells. So this could be from a skin cell or bone or muscle or from nerve. And when you look at the karyotype of this individual, you will see that this individual has 23 pairs of chromosomes. These 23 pairs of chromosomes, one member of the pair comes from the mother, and the other member of the pair comes from the father. The last pair, or the 23rd pair of chromosomes, and most of the time it's not labeled as the 23rd pair, but the 23rd pair of chromosomes are known as the sex chromosomes, and they determine the gender. So if you look at the individual on the left, this is a full complement, is how it referred to. It's a full complement of their genome, their genetic composition, all of the genes on all of the chromosomes, the ones that came from the mom and the ones that came from the dad. And we look at this person, they have inherited an X chromosome and a Y chromosome. That makes them a male. Females inherit X chromosomes from mom and from dad. Now, what we're looking at is this diploid number. Di means two. So how this is referred to, it's referred to as 2N. And the 2N number in humans, in our body cells, the ones that are created in mitosis, is 46. Now, on the other hand, this is the same person, except for this isn't a skin cell. This is a gamete. This is a sex cell. So, since this was a male, then this is a sperm cell, is what we're looking at. So, we're looking at the genetic makeup of the sperm cell. And if you see, it does not have chromosomes in pairs. All that you have is one copy of each type of chromosome, and that includes the sex chromosomes. So males can either donate an X, or the sperm cells can donate a Y. but there will only be one copy in each of them. So these are the cells that are created in meiosis. And if you look at how they're represented, these haploid cells, or I like to pronounce them as haploid, that just kind of helps you remember your math there. This is represented by the N number. So the N number for any species, and it's going to be specific to species, is the base number of chromosomes. How many different types of chromosomes are in that species? So as humans, we have 23 different types of chromosomes. So that is our N number. Now we can play around with this a little bit, and that's what I've done. Up at the top, I have the scientific name for a couple of vegetables. So scientific names are the names of organisms that are universally accepted. They are typically Latin, and they're written in a particular way so that you can distinguish them between common names. So they're usually in italics if you're typing, or if you're writing them out, you would underline them. These two names, they derive from the classification of an organism. So in the room, I have the classification word wall. So going from domain all the way down to species. So domain being the biggest of the classification groups or taxa, but being the least specific. And then going all the way down to species, which they are small groups, they're numerous, and they're most specific. In fact, they're so specific in terms of having shared characteristics that they can successfully sexually reproduce, which means that their offspring can then go on to produce more offspring. So those classification groups or taxa are domain, kingdom, phylum, class, order, family, genus, and then species. So your scientific name, these vegetables, their scientific name comes from the last two of their classification categories, where they fit in based upon their characteristics. Who are they alike? They will share these names. So Solarium is the genus. It comes first because it's first in the classification taxa. It is capitalized, and then Tuberosum is the species. So this is the last of the classification categories. Tuberosum, if you notice, is all lowercase. So what this is representing, this is the scientific name for a potato. And if we look at the scientific name for potato, it kind of makes us think it's a tuber. So you might know about tubers. Tubers grow in the ground. And solarium, it's got something to do with the sun. So it's a plant. It does photosynthesis, so it definitely needs sunlight. Now, looking at chromosome number, the 2N number is 48. So the 2N number is 48. And if I'm to figure out the base number, how many different types of chromosomes that there are in potatoes, all I have to do is find N. So I divide by 2, and the N number is 24. So a potato has more different types of chromosomes than we do. Don't be jealous. Okay, next is Zea maize, and that is the scientific name for corn. And the 2N number for corn is 20, so that means that the N number is 10. Now, the last three organisms here are animals like ourselves. The first one is Rana pippins. Rana pippins. That is the scientific name for the frogs that we will dissect and class for a leopard frog. The N number, so the number of chromosomes, the base number of chromosomes in frogs, there's 13 different types of chromosomes in an egg or a sperm cell. So that means in a skin cell, there's going to be 26. So now to go from the haploid to the diploid, I just multiply by 2. Another example, Felis domesticus. Felis domesticus. So I can see that domesticus and I see Felis. So that is a house cat. And your house cat has 19 different types of chromosomes in an egg or sperm cell. What are you going to find in a skin cell or a whisker cell? You're going to find 38. And then finally, Canis familiaris. So familiaris, Canis. So This is another pet. So this is a dog. This is a domesticated dog. And your dog has 39 different types of chromosomes in an egg or sperm cell. How many are in an ear cell there? 78. With the next part of this, and it's kind of sort of covered up, now it's gone. What I'd like for you to do is to, as the directions say, label each process based on the chromosome number. So you've got choices here. Excuse me, I can't do that in presentation mode. You will drag and drop them to put them where they need to go. I can't get it going. So just drag and drop them where they need to go. Then to the next one, we're going to do a lot of practice with this one because basically when we're done with diploid haploid numbers, we're going to be done with diploid haploid numbers. So I'm going to wear them out for the next day or so. So what I'd like for you to do here is to fill in the chart based upon what's missing. So with some of them, we have the haploid number. You give me the diploid number. Some of them you have the diploid number. Give me the haploid number. And then the last part of this is to write in the text boxes, which I'm not going to click on this because it's going to take me to the next slide, if it is haploid or if it is diploid. Um, Sorry, there is one more slide. And then the last one is just kind of a fill in the blank here. This is very similar to some questions you might see on an SOL test called a closed CLOZE, in which you have a paragraph of information and you fill that paragraph in the same way you would if you were, let's say, taking a test and you had a word bank. This is your word bank, except for here you're going to drag and drop.